wondered why we distinguish life as living and non living things? The fact is, every living organism is made up of cells. No, not that cell. Yeah, this cell. Organisms ranging from this tiny ant to a much tinier microbe to a humongous mammoth. Wait, it is extinct. Yeah, this is right. Humongous elephant. All of these are made up of cells. So typically, all cells can be explained as usual boring statement. They are the fundamental unit of life. They have independent existence or groups that performs vital activities. Next comes the important part, the cell theory. It was put forth by Schleiden and Schwann. Their postulates are all living things are composed of cells and product of cells. All cells arise from the pre-existing cells. Actions of an organism or some of the activities of all the cells in that organism. Okay, now let's get into the main stuff. All cells may be the fundamental unit of life, but not all the cells are similar. Based on their nature, they are classified into prokaryotic and eukaryotic cell. If we see on an evolutionary level perspective, eukaryotes are the most advanced. But if we have a race on cellular activity, prokaryotes would beat eukaryotes by a mile. Let us now get deep into the prokaryotic cells. Basically, all the bacteria, pleuropneumonia-like organisms, blue-green algae comes under prokaryotes. The defining features that differentiates prokaryotes from eukaryotes is that they do not have a well-defined nucleus. Basically, their genetic material is naked, but they have an additional small circular DNA called plasmid DNA. This plasmid DNA is absent in eukaryotes but present in prokaryotes. This plasmid DNA contributes to the antibiotic resistance of the prokaryotes. Most of the prokaryotes have complex cellular outer covering made up of three layers: outer glycocalyx, middle cell wall, and inner plasma membrane. The glycocalyx is made up of mucopolysaccharide. Based on the thickness of the glycocalyx, it can be classified as thin, slimy layer and thick capsule. Inner to the glycocalyx, cell wall is present. Cell wall is made up of a macromolecule called peptidoglycan. Cell wall gives the mechanical strength to the cell. The innermost layer is the plasma membrane, which is selectively permeable in nature. This structure is similar to that of eukaryotes. The plasma membrane sometimes extends into the cell and forms a vesicle-like structure called mesosomes. This mesosome helps in DNA replication, cell wall formation, and many other important things. In addition, prokaryotes may have structures like flagellum, villi, fimbriae for versatile functions from locomotion, food capturing, and reproduction. The plasma membrane encloses an aqueous ground substance called cytoplasm or hyaloplasm. It has ions, amino acids, sugars, nitrogenous bases, enzymes, and other vital cellular molecules. The cytoplasm serves as a medium for all the biochemical processes that happens inside the cell. Next important structure is ribosomes. We might have learned from the schools that prokaryotes have 70s ribosomes and eukaryotes have 80s ribosomes. The 70s ribosome is made of a small 30s subunit and a large 50s subunit. Wait, mathematically 30 plus 50 equals 80. Then how come prokaryotes have 70s ribosomes? Okay, let's unravel this thing. Initially, a complete 70s ribosome is added into the centrifuge tube along with some essential chemicals. Usually, in centrifugation, the result would be having lower weight molecules at the top and the higher weight molecules at the bottom. So, our prokaryotic ribosome would have settled near 70s level. Now, if we split up this 70s ribosome into their respective large and small subunit, they would have sedimented at 50s and 30s level respectively. Okay, now let's get into the main stuff. Ribosomes are made up of proteins and ribonucleic acids at a definite proportion. Their predominant function is protein synthesis. We will be seeing a separate lecture for protein synthesis in the future lecture sessions. Sometimes ribosomes may get attached to the messenger RNA and forms a complex called polyribosomes or polysomes. Next organelle is the inclusion bodies. These are the storehouses of the cells. They store variety of food substances, wastes, and other cellular metabolites. Sometimes they form a gaseous vacuoles in order to float in water. Apart from all these characteristics, prokaryotes lack membrane-bound organelles such as endoplasmic reticulum, Golgi bodies, centrioles, mitochondria, and chloroplast. But bacteria do have a light harvesting complex called chlorosomes that are scattered throughout the cytoplasm. It helps in photosynthesis in prokaryotes. Furthermore, mesosomes helps in cellular respiration as it contains enzymes that are similar to the eukaryotic mitochondria. Prokaryotes have different mode of nutrition from photoautotropic, chemoautotropic, and chemoheterotropic. Some of the bacteria even have a symbiotic association with higher plants. These bacteria fix atmospheric nitrogen to the plants and in return, plant gives protection and nutrition to the prokaryotes. This type of symbiotic association is called mutualism. Bacteria reproduce both sexually as well as asexually. In asexual reproduction, they reproduce by binary fission. In this process, the whole bacteria divides into two identical cells by fission. The resultant organisms will be having similar genetic materials. In sexual reproduction, it happens by conjugation. In this process, the pili or the flagella forms a bridge between adjacent bacteria and they transfer their plasmid genetic material with each other. Thus, conjugation results in recombination of genetic material. That's all about the structure of prokaryotes. Thank you, friends. See you soon in the next video.